Hello, BISC1323. This is the beginning of recorded lecture 1-1. Because this course is a continuation of BISC1303, we're not starting with chapter 1, we're starting in chapter 20, Phylogenies and the History of Life. So this is kind of an organizational big picture chapter before we get into the specifics of things after this. Our first objective here is supposed to be organizing life on Earth. This brings us to an important term, taxonomy. Taxonomy is simply the science of classifying organisms. So this involves naming organisms. This also involves organizing organisms, putting them into some sort of groups with one another. Ideally, the way we want to classify things is based on their evolutionary history. We want to indicate things that share a close common ancestor. It should be grouped closely to one another, and things that are distantly related evolutionarily are grouped distantly from one another. If this is sounding very abstract, this is called phylogeny. It's okay. We've got cool pictures that diagram this evolutionary history, these phylogenies. So we diagram this in things called phylogenetic trees. Here's a generic looking one that just shows organism 1, 2, 3, 4, 5, but we can see there are groupings here. And 1, for example, whatever this organism or group of organisms is closely related to 2 and 3. They share a common ancestor down here. These sort of branch points are speciation points. And group 1 is distantly related to group 8. I guess they share a common ancestor if you go back really far, but again, the distance on this sort of tree indicates evolutionary relatedness. For one that's not just 1, 2, 3, 4, 5, 6, 7, 8, here's a preview of a later chapter. We will talk about vertebrates, fish and amphibians and mammals and reptiles and birds. And yeah, we can see this sort of played out in a real phylogenetic tree. So there's a very important term when it comes to talking about phylogenetic trees, and that is the term clades. Something we call a clade is a complete branch. This includes an ancestor and all of its descendants, and it's also known as a monophyletic group. Now, these clades can be big or these clades can be small, as long as it's something including an ancestor and all of its descendants, we can call it a clade. So to go back to this one, 1 is a clade. 2 is a clade. Each of these individual small branches, those are clades. 2 and 3 together are a clade. If we have an ancestor here, including all of its descendants, that's a clade. 1, 2, and 3 are part of a clade as well. If we go back to this branching off point, this includes an ancestor and all of its descendants, 1, 2, 3. 4 and 5 make up a clade, 7 and 8 make up a clade. If you wanted to do a big one, 4, 5, 6, and 7 and 8 are all part of a clade. So what is not a clade? Well, something that's not a clade is, let's go back to this vertebrate phylogeny here. So something that's not a clade is reptilia. So we all know what reptiles are, but if we draw a circle sort of around reptiles, we can see, turtles, lizards, snakes, crocodiles, but not birds. Birds are not reptiles. This is what reptiles are. That is an ancestor and a lot of its descendants, but it doesn't include birds. We're excluding birds from the group of reptiles. This is not a monophyletic group. So again, it's supposed to be based on clades. Something like reptile is a traditional group, and it's definitely not a clade. What we're calling things like this is a paraphyletic group, as opposed to a monophyletic group. Remember, that's another word for clade. Paraphyletic groups are a clade minus a subgroup or 2 or 3. In this case, this would normally be a clade, but we're excluding birds from this term of reptiles. So yeah, paraphyletic. There is one more term to describe groups here, and that is the term polyphyletic group. So if we were to lump mammals and birds together, why would we lump mammals and birds together? Well, we're both warm-blooded. And so, yeah, it would make sense to create some group of warm-blooded vertebrates here, including mammals and birds. But as we can see, they're distantly related to one another. This is not an evolutionary relationship here. We call this a polyphyletic group. We're not just doing a clade and subtracting, one or two groups. Polyphyletic means just two completely different things that happen to be grouped together. Polyphyletic group, a set of organisms not directly evolutionarily related. Now, taxonomy was about organizing things. And yeah, we're going to see a lot of these phylogenetic trees, not just in the vertebrates chapter, but in other chapters as well. Taxonomy is also about naming things. And as part of this, there's a taxonomic classification system, which has at least 8 levels of organization. These levels are called taxa, or taxon is singular. So I think the best way to look at these 8 is to walk through an example. You don't need to memorize this particular example, but you do need to know these 8 levels. We're going to start with the biggest level, the most inclusive level. In this particular example, the taxon here at the very top is called domain. We are in domain eukarya, and you should remember what eukaryotes are from BISC1323. Eukaryotes are animals and plants and fungi, and we didn't really talk about protists in that class, but you better bet we'll talk about protists in this class. But yeah, domain eukarya includes a bunch of stuff. After domain is the taxon kingdom. So now we're getting a little more specific, a little more exclusive. Animalia includes animals, vertebrates, invertebrates, but we're not talking about plants or fungi or protists anymore. To go more specific, after kingdom, there is phylum. In this case, phylum chordata, something with a dorsal nerve cord. That's a feature we'll talk about later in the quarter. After this is, subphylum. So I did say, to go back here, that there were at least 8 levels of organization. We'll see many other examples throughout the quarter. Sometimes there are superphyla or subphyla or subclasses, or sometimes there are definitely more than 8, but we got to know the basic 8 here to start with. In this case, there is a subphylum. Vertebrata, we're talking about vertebrates, something with a skeleton, with vertebrae. After phylum, there's class. In this case, we're in class Mammalia, mammals, kind of a skunk, a panda, some sort of monkey here. After class, there is order. In this particular example, it's order Rodentia, so we're talking about rodents, mice, and rats. After order, there is family. In this case, it's Sciuridae, so we've got chipmunks and squirrels. After family, there is genus, Sciurus, that's squirrels. And finally, species is the most specific taxon. It's Carolinensis here. This is the eastern gray squirrel. That's the exact species of squirrel that you see scampering around Tech Campus. So to review, from the biggest, most inclusive to the smallest, most exclusive, we've got domain, kingdom, phylum, class, order, family, genus, species, and noting sometimes there are subtaxa. I think the best way to remember this is with a mnemonic fitting the structure of D, K, P, C, O, F, G, S. In my opinion, the best mnemonic for remembering this is one that you make up yourself. Sorry, you got a little work to do, but you can Google this if you're really lazy. I'm sure there are other mnemonics out there, but yeah, come up with one that's weird, that's like specific, that's personal. If you come up with your own mnemonic for D, K, P, C, O, F, G, S, you'll remember it a lot better than something you randomly find on the internet. That's just my advice, though. One other thing about the 8 taxa. So when it comes to these final 2, the most specific 2, genus and species, we can put those 2 together to make up an organism's 2-word name. Every organism out there has a special 2-word name. We call this binomial nomenclature. There are a couple of rules associated with this. When we're writing out an organism's binomial nomenclature name, it's always in italics or underlined. Usually underlined is just if you're doing handwriting, because it's hard to do italics in handwriting, but if you're seeing anything sort of typed or printed out, it's going to be in italics, just to let you know we're talking about something's two-word name. The other rule is that the first word, the genus, is always capitalized, and the second word, the species, is never capitalized, regardless of what spell check likes to do with E. coli. The E. coli would be a good example of this, Escherichia coli, and another good example is Homo sapiens. That's us, that's humans. We're in genus Homo, species sapiens, our two-word name, Homo sapiens. Now we're going to go through different kingdoms and phyla and classes and stuff as we make our way through the quarter. But right up top in this chapter, we can talk about all the domains because there are only 3 of them. So you should know that all living things are contained within 3 of these sort of top level, big organizational taxa, the domain level. There's domain bacteria, domain archaea, and domain eukarya. And hey, here's another phylogenetic tree showing, color-coded for your convenience. There's bacteria, that is a clade. There's archaea, that is a clade. And there is eukarya, that is a clade as well. I like the little "you are here" star in animals. So yeah, we'll talk about each of these in their own chapters. But yeah, just wanted to point out, these are the 3 domains of life. Now, an important question you might be having right now is, how do we know this? How do we make these? How do we know that Methanobacterium is, more closely related to the halophiles than it is to Thermoproteus or any of these other things? How do we construct these phylogenetic trees? How do we determine what these evolutionary relationships are? Well, there are a couple of ways to do this. There's the old school way and there's the more modern way. Let me talk about the old school way first. One way to do this is to assume or infer that organisms with similar features have them because they share a common ancestor. So these relationships can be inferred based on what are called homologous traits, which are also known as synapomorphies. It's just a couple of fancy words and phrases for a shared physical feature. So to give you an example of this, let's look at some arm and hand bones across some vertebrates, human, dog, bird, whale, very different animals doing very different things with their arms, flying, swimming, walking around on their hands, using their hands to grab things. But all of these structures have the same basic bones. They have the same basic radius and ulna and wrist bones and digits, and they're just modified to fulfill different functions. Seeing all of these different animals sharing this common feature can make us assume that they all have this hand, this arm, because they share a common ancestor that had an arm like this. And yep, we'll talk about this example in more detail when we get to our vertebrates chapter, but indeed there is an ancient tetrapod that started this whole thing. And so there is a common ancestor between all of these because of this shared feature. So we put together these phylogenetic trees based on, looking at physical features that different things have. Again, the assumption being organisms with similar traits have them due to inheriting them from a common ancestor. However, sometimes this doesn't work out all that well. Comparison of physical traits can sometimes be confusing due to what are called analogous traits. Analogous traits, as opposed to homologous traits, analogous traits are traits that fulfill a similar function but are structurally and evolutionarily different. So what do I mean by this? Well, let's look at another example. Wings. These are butterfly wings. These are bat wings. They have a lot of similarities. These are paired appendages. They have a lot of surface area and are very flat and thin. which helps with, the whole flying thing. And yeah, they both fulfill the same function of allowing these butterflies and these bats to fly. So at first glance, you may say, oh, this is a shared common feature. They must have a common ancestor, just like these homologous traits. But you have to look more closely. If you look more closely at this feature, you can see that the bat wings have bones and cartilage, and there are blood vessels in here and skin. But if you look more closely at the butterfly wings, these are made of chitinized protein, and they're connected to a circulatory system of hemolymph, and they're built completely differently, even though they're fulfilling the same function. So this is, and sometimes it's not always cut and dry, whether something is an analogous trait or a homologous trait. This is why I said it can sometimes be confusing to determine whether a shared feature it comes from a different evolutionary lineage or whether it's because of a common ancestor. So yeah, I'm admitting that this is a problem, but yeah, analogous traits, similar function, but they're structurally and evolutionarily different. Homologous traits based on a common ancestor. And yeah, analogous traits can be confused for homologous traits, but again, they're not due to a common ancestor. These butterflies and these bats evolved their wings independently of one another. And this actually happens a lot of different groups of organisms evolving a similar body feature or capability independently of one another, coming up with the same idea separately. This happens so often, we have an important term for this. It's called convergent evolution. I've got my key terms here. Remember, when things are highlighted in the notes, it's something that you'll find in the key terms document. Convergent evolution is defined as the process by which groups of organisms independently evolve to similar forms. And yeah, a lot of groups have evolved wings, but here's another example. These both look like big cats. This is Smilodon up here. This is Thylacosmilus down here. If you were to look at these side by side, you'd probably guess that these are closely related to one another, that they're all sort of closely related to living tigers, lions, cats. But if you look at them more closely, this thing down here is, well, it's not technically a marsupial, it's a metatherian. It's like a marsupial. This thing down here is more closely related to a koala than it is to a tiger or a lion or any cat at all. This is, a sort of proto-marsupial that has evolved this cat-like body due to convergent evolution. Two very different groups of mammals coming up with the same body plan. Again, the strong forearms, the big mouth, the saber fangs here, because it works. This is a really great way to be an apex predator and to take down large prey with these giant canines. And again, looking at an evolutionary tree, Smilodon, closely related to cats, but this Thylacosmilus, it's over here with kangaroos and koalas and stuff. Origin of saber teeth happened separately in different groups. That's convergent evolution. One more example, because again, we're going to see this a lot throughout the quarter. Don't sweat all the crazy details here, although you can read them if you're curious. This is a dolphin, which is a mammal. This is an ichthyosaur, which is a extinct reptile. Not a dinosaur, but a reptile. And they have a lot in common. They've got the dorsal fin. They gave live birth the same way. They have similarly shaped skulls, similarly shaped limbs. They've got flukes here on their tails. And they're not closely related. Again, this is a reptile. This is a mammal. But they evolved very similar looking bodies because it works. If you're going to be a large marine predator and you're eating fish, this is what peak performance looks like. And if we want to look at their tree, yep, here the ichthyosaurs are closer to lizards, and here's the common dolphin over here, closer to elephants and stuff. Definitely far apart from reptiles. So yeah, we'll see many more examples, but convergent evolution. This is due to similar selective pressures on these different groups. Again, the selective pressure to take down large prey, to survive in the open ocean, it's going to shape different groups to have very similar looking bodies. Now, All of this comparison of physical features was the old school way of doing things. Modern taxonomy is based on computational analysis of gene and protein sequences. Not as visually interesting to examine, but it looks like this, of taking a protein sequence of different organisms, different species, and lining them up and saying, hey, they've got a lot in common with one another. Obviously, human, mouse, rat, cow, and chimp, these are all mammals, so they should have a lot in common with one another, but we can start to see some differences in this position. Most of them have an alanine amino acid, but it looks like humans and chimps have a serine amino acid. Oh, humans, chimps, more closely related to one another than they are to rats or cows? Who'd have thunk it? And we can continue to scan through here. Yep, here's another difference that humans and chimps have in common with one another, different from these others. There's another difference. Oh, here's one that's different in cows. There it is. That's different in cows, but everyone else has the same. And so this is the sort of analysis, this sort of comparison that a computer has to do. Again, a computer is going to scan through these long sequences and determine how closely or distantly related they are based on how similar or dissimilar these gene or protein sequences look to one another. Importantly, this is how we construct phylogenetic trees for things like microbes that don't have a lot of obvious, physical body features that we can find. We can analyze the DNA of these microorganisms and construct phylogenetic trees for them. This is not without its problems as well. So it makes it sound pretty cut and dry and easy just to compare them. It has problems, something called horizontal gene transfer. So the assumption we have been making with all of this is that we inherit the genes that we have from our ancestors. A parent passes genes down to offspring, and they pass them down to their offspring, and they pass them down to their offspring. And if things have a similar gene sequence or a similar physical structure, it's because they had some ancestor that passed those genes down to them. Vertical gene transfer from ancestors to descendants. But what can also happen is horizontal gene transfer. The key terms define horizontal gene transfer as the transfer of genes between unrelated species. So to look at an example of this, here are some aphids. These are bugs, the scourge of many a gardener out there, stealing nutrients from these plants. Usually they're green. These ones have sort of a rusty, brownish red coloration to them. And if you look closely at their gene sequences, at the protein sequences of these aphids, that pigment doesn't look like an insect pigment, because there are other, closely related insects to aphids that have a coloration similar to this, but that's not a bug pigment. This reddish brown pigment is very similar to a gene and a protein that fungus makes. So, whoa, is this convergent evolution? Is this, a completely separate lineage of bugs that evolved from fungus that look exactly like other aphids, but they've got this fungus gene? No, it's not convergent evolution. They got this fungus gene by living in very close association with fungus that also, attacks these plants. And genes just jumping from one species to another doesn't happen very often in animals, but it happened here. That's what horizontal gene transfer is. It got this pigment gene not from its ancestor, but from something else, from a fungus in its environment. And so, again, that makes this difficult to do. Doesn't happen often in vertebrates or even in invertebrates, but happens all the time in bacteria and archaea, which is going to make trying to line up their sequences and construct these trees very challenging. But Again, despite these challenges, we still construct these trees. We still use these trees. We're going to see a lot of them throughout this quarter. This is the end of recorded lecture 1-1. Again, just sort of a short intro to some terminology here. We will get more into some specifics in the next one.